man stands at a lightbox in a basement in Copenhagen, holding 2 photographs of human eyes between his fingers. It is 2008. The fluorescent tubes overhead hum at the pitch they always hum at, a low electric note that anyone who has worked in a university genetics lab will recognize instantly. The room you are picturing smells of cold plastic and old coffee. Behind him, behind you, a centrifuge winds down with a soft whine. He moves the 2 photographs closer together. One iris is brown, the other is blue. He places them side by side on the lit glass, and he looks down at a printed chromosomal readout taped beside them. And somewhere in the long string of letters, his finger finds the position he came down here to confirm. One letter, a single base on a single strand, sitting 21,152 pairs upstream of a gene called OCA2. He marks it with a soft pencil. The lightbox glows. The photographs glow. The man, whose name is Hans Eiberg, the one you are watching, has just confirmed something that almost no one outside this room knows yet. He has just confirmed that every single blue-eyed person on Earth, every one of them, from Reykjavik to Kabul to Istanbul to a farm in Iowa to a small town in Tasmania, is descended from one common ancestor, not metaphorically, genetically. One person, alive somewhere in the late Stone Age, sometime between 6,000 and 10,000 years ago, near what is most likely the northwestern shore of the Black Sea. One iris that scattered light differently, one child, one night. And every blue eye that has ever opened anywhere on this planet, in every century since, traces back to that single moment in one body. You think blue eyes are just a common trait in Northern Europe. They are not. They are the longest, quietest founder effect in the human genome, a tiny mutation in a gene that does not make blue pigment, in a place that should not contain it. Written on the chromosome of one stranger, copied a few 100 million times, and arriving here, tonight, in eyes that may or may not be your own. This is the story of that mutation. It is going to take you and me about 2 hours. There is no rush. Make yourself comfortable. The chapter you are listening to opens in a Danish laboratory and ends in an empty room in the dark. And between those 2 points, we will travel through caves in Somerset and Iberia, through grasslands in Ukraine, through lecture theatres and Mesolithic estuaries, and the long, strange detour of a single guanine base inserting itself into the human genome and refusing to leave. There will be physics, there will be skin, there will be cheddar man, there will be a pastoralist driving cattle west across a steppe that no longer exists in any recognizable form. There will be a question we cannot yet answer, even with all the genome sequencing in the world, about why this mutation spread at all. Settle into your own breath. Let your shoulders drop. The room is warm, the night is long, and you and I have plenty of time to walk this slowly. Eiberg is still at the lightbox. The 2 photographs are still under his finger. The chromosomal readout is still taped beside them. To understand what he is looking at, you have to understand something strange about blue eyes that almost everyone gets wrong, including most of the people who have them. There is no blue pigment in the human body, including yours. None. Anywhere. Not in the iris, not in the skin, not in the hair. Nowhere. Whatever color you call your eyes, they do not contain that color as a substance. They contain melanin, which is brown to black, in varying amounts, and they contain a translucent layer of stromal tissue at the front, and they contain a dark, pigmented epithelium at the back. That is everything. There is no blue chemical. There is no blue dye. There is no blue molecule. The blue you see, when you see it, is a trick. It is the same trick, you may already know, that makes the sky blue at midday. It is structural, not pigmentary. It is light scattering off small things. And the only reason any human iris ever appears blue is because somewhere upstream of one specific gene, a switch has been flipped that tells the front of the iris to stop making melanin. Once melanin is gone from the front layer, the back layer is still dark, and white light entering the eye scatters off microscopic collagen fibers at the front. And the short blue wavelengths bounce back out toward the observer, and the long red wavelengths pass through and are absorbed by the dark layer behind. And what comes back to the observer is blue. Not blue pigment, blue physics, the same physics that paints the sky. Eiberg knows this. The community knows this. The strange part, the part that has been hidden inside the genome for the better part of a century. The part that he came down to the lab tonight to nail down is what flips the switch. Where, exactly, is the instruction that tells the iris to stop making melanin in the front layer? Which letter, on which chromosome, in which gene? He found it. The team found it. They have been working on this for years. It is a single nucleotide polymorphism, which is the technical term for one base out of place. It sits at a position you can now label, with the scientists, as RS129138332. It is in a gene called HERC2, in the 86th intron, the long stretch of non-coding DNA that lies between the protein-making sections. Most people, perhaps you among them, when they hear that a gene controls something, picture the gene itself doing the work. But that is not what is happening here. The gene that suppresses melanin in the iris is not HERC2, it is OCA2, the neighbor, sitting just downstream on the same chromosome. OCA2 makes a protein called P, you should know, which regulates the acidity of the little organelles where melanin is built. When OCA2 is fully knocked out, you get albinism. When OCA2 is intact, you get pigment. What RS129138332 does is something far more delicate. It does not break OCA2. It just dials it down, in one tissue, in one layer of one organ, in one part of the body. The rest of the body keeps making melanin normally, as your body would. The skin, the hair, the back of the iris, the moles on the back of your hand, all of it carries on as if nothing has happened. Only the front of the iris is told, by the regulatory element, 21,152 base pairs upstream, to make a little less of the protein that makes the pigment that absorbs the light. That is all. That is the whole genetic story. One letter, in one regulatory site, in one direction, in one tissue. Hold that picture in your mind. Eiberg's pencil mark on the lightbox, the one you can see, is the position. The two photographs in his fingers, in front of you, are the result. What made the team hold their breath for the year before this confirmation came back was something they had begun to notice in the data and could not quite believe. Every blue-eyed person they had sampled, every one, carried not just the same mutation, but the same long stretch of DNA on either side of it, the same haplotype, the same neighborhood. They had run the test on 100 Danish trios from the Copenhagen Family Bank, the standard linkage mapping tool of Northern European genetics. They had run it on 155 blue-eyed Danes pulled from a population study. And then, because the result had begun to look almost suspicious in its cleanness, they had reached out for samples further afield, 5 blue-eyed people from Turkey, 2 from Jordan, the kind of population that, on the rare occasion blue eyes appear there, appears against a backdrop of overwhelmingly brown-eyed neighbors. If the mutation had occurred independently in different populations, parallel evolution, the same change happening twice, the haplotype would differ. The neighborhood around the mutation would carry the marks of separate descent. It does not. The Turks match the Danes. The Jordanians match the Turks. Everyone matches everyone. The flanking DNA is identical. The neighborhood is shared. There is only one neighborhood. This is what a founder effect looks like in a genome. You should hold the term. When a single individual carries a mutation and their descendants spread, and over time the random shuffling of recombination chews away at the original neighborhood. The further you get from the founder, in time and in generations, the less of the original flanking DNA you find. The fact that the flanking DNA, in this case, has barely been chewed at all means the founder is recent. The fact that it is identical across continents means there has only ever been one founder. The mutation, the one that lives in your story or someone you love, did not happen many times in many places. It happened once. Eiberg taps the chromosomal readout under his finger and stands back from the lightbox, and the basement is quiet apart from the centrifuge, and somewhere on the floor above someone is closing a door. He has just confirmed descent from the Copenhagen family bank, 100 trios, 155 Danes, 5 Turks, 2 Jordanians, and every single one of them, on this single position on chromosome 15, is reading from the same library card you might be reading from now. Now, take a breath. Let the lab fade for a moment. The walls are concrete. The fluorescents hum. The 2 photographs lie on the lightbox. Hans Eiberg will write up the paper. The paper will appear, you can look it up, in a journal called Human Genetics in 2008. The world will read it slowly, the way it reads such things, and over the next decade the implications will deepen rather than weaken. Every new genome that comes out of every new ancient cave will be tested at this position, and every test will confirm what the lightbox confirmed tonight. 1 mutation, 1 founder, 1 inheritance. We are going to leave Eiberg here for now and walk slowly back through the genetics of how this all works. Because the next thing you have to understand is the strangeness of the molecular switch itself. You and I are going to go to a lecture theatre, the kind you may remember from school, with tiered wooden benches and a single overhead projector. The kind where the lights dim and the projector hums, and a diagram appears on a screen that is older than most of the students. The diagram in front of you shows 2 genes, side by side, sitting next to each other on chromosome 15, OCA2 and HERC2. The lecturer is older than the projector. He taps the diagram, in your line of sight, with a wooden pointer, in a place that looks empty. He says, "This part, this is intron 86. There is nothing here that codes for anything. It is non-coding DNA. Junk, the textbooks used to call it. Filler, spacer. Nothing." He says, "It is not nothing." He says, "A single letter in this empty stretch is the difference between a brown eye and a blue one in nearly every human being now alive." The lecture theatre is quiet. The projector hums. The pointer rests on a place on the diagram where, 20 years ago, no one would have stopped to look. 20 years ago, before you were perhaps thinking about any of this, the diagram would have been simpler. There would have been the gene, OCA2, the one that codes for the P protein, the one that controls the pigment. Around it would have been the long, dull stretches of non-coding DNA you may have been told were scaffolding. Then we learned, slowly, painfully, by accident in some places and by careful design in others, that the non-coding stretches are where most of the regulation happens. Not the gene itself, the neighborhood around the gene, the promoter, the enhancers, the little switches that say turn this on, and the little switches that say turn this off, and the little switches that say turn this on but only in this tissue, only at this temperature, only when the body is making this hormone. The gene is the instrument. The non-coding region is the conductor, and 1 letter in the conductor's score, in this particular case, tells your iris, or someone's iris, to play softer. The mechanism, when you hold it in mind, is precise enough to be slightly miraculous. The OCA2 gene is making P protein. The P protein, you can imagine it, is regulating the pH of melanosomes. The melanosomes are taking melanin precursors and turning them into the brown pigment that fills the front of the iris. All of that is happening normally. None of it is broken. What Rs129138832 does is sit in a regulatory site upstream and decide, in cells of the anterior iris stroma, how much OCA2 transcription gets to happen at all. With the ancestral letter adenine, the regulatory site binds the proteins it is supposed to bind. The gene transcribes at full strength. The P protein gets made at full strength. The melanosomes acidify properly, the melanin precursors process properly, and the front of the iris fills with brown. With the derived letter guanine, G, the regulatory site binds those proteins less efficiently. The gene transcribes at lower strength. P protein gets made in smaller amounts. The melanosomes do not acidify as well. Fewer precursors get processed. Less melanin ends up in the front of the iris. The back of the iris, governed by a different regulatory wiring, keeps making melanin as it always has. The front goes pale. And once the front goes pale, structural physics, the kind you do not need to think about, takes over. The color is no longer absorbed at the front. It is scattered there, in front of you. The Tyndall effect, named after the 19th century Irish physicist who first described why a flask of murky water turns blue when you shine a light through it, kicks in. Short wavelengths bounce back. Long wavelengths sink in. The eye looks blue. This is why blue eyes, perhaps yours, shift hue with the light. A brown eye is brown because melanin is absorbing all the wavelengths and reflecting back the long ones. The color is fixed by the pigment, and the pigment is fixed by the cell. A blue eye is blue because there is no pigment doing the work. The color is being made on the fly. In real time, by whatever ambient light is in the room scattering off the structure of the stroma. Bring a blue eye into bright sunlight, and you will see it goes brilliant cyan. Take it into a candle-lit room, and you will see it goes grayish slate. Wave a torch across it, in your own hand, from the side, and you will see the color shift as the angle of incidence changes. None of that happens in a brown eye. The pigment fixes the color. The structure does not. This is also why babies are so often born with blue or gray eyes that change in the first 1 to 3 years of life. Melanin production in the iris, you should know, is partly stimulated by light. An infant emerging from the dark of the womb, perhaps you can remember holding one, has not yet built up the pigment it will eventually carry. The front of the iris is translucent because the cells have not yet finished their work, not because RS12913832 has switched anything off. Over the months and years that follow, in children who do not carry the homozygous G genotype, the cells deposit melanin gradually into the stroma and the eye darkens. By the 3rd birthday, in most cases, the color has settled into whatever your genetic instructions dictate. In children who do carry G, both copies of the chromosome reading the suppressed regulatory site, the cells never deposit enough melanin to obscure the structural blue, and the eye stays the color it was at birth. The first photograph of a blue-eyed adult is, in a sense, a continuous line drawn back to the first photograph of that same person as a newborn. The melanin never came in. The structure was never overwritten. The eye, in a small but real sense, never grew up, and yours, whatever color, holds its own version of that history. Eiberg's team did not stop at the haplotype map. They wanted to prove, and you can follow them in this, that the G allele actually reduced OCA2 transcription, not just that it correlated with blue eyes. They ran an electrophoretic mobility shift assay, the standard biochemical test for whether a piece of DNA binds to a regulatory protein. The G version, as you would expect by now, bound less efficiently than the A version. They ran a luciferase assay in cell culture, which is a way of attaching a glow-producing gene to the regulatory site and measuring how much glow you get. The G version glowed dimmer. The biochemistry confirmed the genetics. The regulatory site, with the derived letter, really does dial down the gene. You can take that as fact. The mechanism was no longer a guess. It was a measurement. It was a number on a meter you can almost see, in a Danish lab, in 2008. You should know, and this is the kind of detail the lecture theatre's older listeners will pause on, that this is not the only gene involved in human eye color. It is the largest single contributor by far. It accounts, you should know, for the majority of the variation in the trait across European populations. But the full picture, the one you should hold, is that human eye color is polygenic. There are at least 16 genes that have been confirmed to make some contribution to the color of the iris, and probably more that we have not yet identified. There are modifier genes that brighten or darken. There are genes that contribute to green or hazel or amber. There are genes that determine the patterning of pigment, the rays around the pupil, the slight variations in the stroma, the Mendelian high school version of eye color, where brown is dominant and blue is recessive, and a Punnett square will tell you exactly what your children's eyes will look like. That version is a teaching simplification. It is true, you may already sense, in broad strokes, and it is wrong in the details. Two blue-eyed parents can, occasionally, rarely, have a brown-eyed child. Your old textbook quietly skipped over that. The genetics are not that clean. They never were. But the point of all of that, for the purpose of this story, is that none of those other genes carry a founder effect signal, anything like the one at RS129-13832. The other genes have variants that have arisen independently across many lineages, in many places, at many times. They show, when you look at them, the kind of genetic patchwork that real human populations show. Only this one site, this one letter, in this one regulatory neighborhood, shows the unbroken signature of single source descent. Only this one site is the founder mutation. The others are noise around the signal. The signal itself, the one you have been listening to, is one ancestor, one mutation, one inheritance. Now, there is a small caveat to address before we move on, because the dossier flags it, and because it would be dishonest to leave it out. The dating of this mutation 6 to 10,000 years ago, somewhere near the northwestern Black Sea, is the consensus view. But it is not the only view, and the picture has become slightly more complicated as the ancient DNA record has deepened. There is a body of work suggesting that the molecular precursors for light pigmentation, including some of the variants involved in lighter skin and possibly traces of the kind of regulatory shift seen at RS129-13832, may have been circulating in low frequency among ancient North Eurasians, a deep population from the Upper Paleolithic, far older than the Neolithic transition, far older than agriculture, far older than the Black Sea timeframe. The strict reading of one mutation 10,000 years ago in one place may turn out to be a slight oversimplification, in the way that all clean stories about evolution turn out to be. The signal at RS129-13832 is real. The founder effect is real. The single ancestor is real. But the full timeline of when that ancestor lived and what other faint pigmentation variants were drifting around the human gene pool in the deep past may shift as the data thickens. We will return to this, you and I, when we talk about the great westward migrations. For now, hold the date loosely. 6,000 years, 10,000 years. Somewhere in there, somewhere near the Black Sea, somewhere in a body that is no longer in the world, and you can feel that, the long absence. Let the lecture theatre dim. Let the projector cool. The pointer leans against the wall. The lights go out. Around you, one row at a time. The diagram fades on the screen, and the screen fades into the dark wall behind it, and the lecture theatre exists, like all such rooms, only in the time you are sitting in it. You and I are going to walk now from the diagram to the body. We are going to go from chromosome 15, abstract, suspended on a slide, to a particular cave in Somerset. In 1903, on a wet afternoon, where a labourer's pickaxe has just caught against something pale in the limestone, the cave is called Gough's Cave. The labourer, in the picture you are holding, does not know what he has hit. The skull he is about to lift, slowly, with the particular care that men of that century used when handling something that felt important, is more than 9,000 years old. Its owner has been dead since long before the pyramids were built, since before Stonehenge was raised, since before agriculture existed in the British Isles in any recognizable form. His skin was dark, his hair was dark, His eyes, and this is the part that no one in 1903 could possibly have known, were blue. Not pale grey, not light hazel. Blue, the same scattered light, the same Tyndall effect, the same suppressed front stroma melanin in a face that, by every assumption modern Europe would carry into the 20th century, ought not to have been able to look out of blue eyes at all. His name will become Cheddar Man. The cave you are walking toward is a few hundred meters outside the village of Cheddar, where the cheese comes from. The limestone in this part of Somerset has been forming for hundreds of millions of years, holding water through it like a slow filter, and during the last ice age, when most of Britain was a tundra walked by reindeer, the cave was a refuge for a small population of hunter-gatherers who travelled along the river systems of southern England. The skull, in your mind's eye, will be measured, photographed, written about, and stored. It will sit in a museum case in the Natural History Museum in London for the next century. Mostly ignored, perhaps you have walked past it. Its DNA will not be sequenced until 2018, more than 100 years after the labourer first lifted it. When the team at the Natural History Museum and University College London finally do the sequencing, they will use a technique called petrous bone sampling, drilling into the dense bone surrounding the inner ear, which preserves DNA better than any other part of the skeleton. They will extract, and you can imagine the painstaking work, a few billion fragmented base pairs of ancient genome. They will reassemble them, slowly, with all the modern bioinformatics that did not exist in 1903. The work could not have existed anywhere except in the kind of computing environment that took the better part of the 20th century to build. They will look at the SLC24A5 locus, which carries one of the major variants for European light skin. Cheddar Man has the ancestral allele, dark skin. They will look at SLC45A2, the other major light skin locus, ancestral allele, dark skin. They will look at MC1R, which contributes to the dark versus light hair axis, ancestral, dark hair. They will look at RS12913832, the position Eiberg pinned down in the basement in 2008. Derived allele, blue eyes. Take that picture in for a moment. A man, fully grown, walking the chalk paths of southern England 9,000 years before the present, with you watching him from much later. His skin is dark, his hair is dark, his face is the kind of face that, transplanted to modern Britain, would turn heads in a way it never would have turned heads in his own lifetime. Because in his own lifetime he was simply one of the local hunter-gatherers, walking the local paths, and his neighbours looked very much like him, and his eyes were blue. Pale, scattered, sky blue, as the dossier puts it, in a body built for the long latitudes of northern Europe, but not yet bleached by them. The textbooks of the 20th century, written before the ancient DNA work was possible, had put light skin and light eyes into the same evolutionary basket, on the assumption that they had co-evolved as humans moved north. Cheddar Man, on his cold morning in Somerset, walks straight through that assumption and out the other side. The basket is wrong. The traits are not coupled. The eyes were here. You should hold this, long before the skin was. Dark hunter-gatherers walked Britain for thousands of years, carrying a Tyndall blue gaze that almost no one in 1903 would have thought possible. He is not alone. He is not even rare. Every Mesolithic genome that has come out of Western European caves in the last decade has shown some version of the same combination. La Braña 1, recovered from a cave in Leon, in northern Spain, dated to around 7,000 years before present. Sequenced in 2014 by Carles Lalueza Fox and Inigo Olalde at the Institute of Evolutionary Biology in Barcelona, has the ancestral alleles at the skin lightening loci and the derived G allele at RS12913832, dark skin, blue eyes. Lochbohr Man, recovered in Luxembourg, dated to around 8,000 years before present, sequenced in the same wave of work, shows the same pattern. dark skin, blue eyes. Slightly to the north, the Motala hunter-gatherers from Sweden, also around 8,000 years old, show the first hints of the skin-lightening transition beginning to creep into the genomes of European populations. Alongside the same blue-eye allele that has by now traveled the breadth of the continent in its dark-skinned hosts. The Western hunter-gatherer cluster, as the geneticists now call it, was a continent-spanning population of dark-skinned, blue-eyed people who lived in Europe from before the last ice age receded until well into the agricultural transition. They were not a footnote. They were, you should remember, the dominant population of their time and place. They were, for thousands of years, the face of Europe, not the face you have been carrying in your imagination. And they are not the face anyone in modern Europe, including perhaps you, carries any longer in their imagination. The dissonance is sharper if you place yourself for a moment in their position. Stand in your body, in your own skin, in your own room. The walls around you are warm. Now hold the thought of those walls and let them dissolve, and put yourself instead at the edge of an estuary on a grey morning in the early Holocene. The Atlantic light is silver. The wind carries, to you, the smell of mud flats and salt and decaying reeds. The ground under your feet is cold. You are not alone. There are other people around you, dressed in skins, dark-haired, dark-skinned, with the kind of muscular leanness that comes from a life of hunting and walking and gathering. Their faces look out at the water. Some of them have brown eyes. Most of them do. But the man standing closest to you, the one with the spear resting against his shoulder, has eyes the color of the sky behind a thinning cloud. He is not unusual. His mother had them. His grandmother had them. The trait runs, you should picture it, through his family and through several other families along the river. Where the trait came from, no one in the camp will ever know, because the language to ask the question does not exist. Because the technology to answer the question will not exist for 9,000 years. Because the man on the lightbox in Copenhagen has not yet been born. The eyes are simply there. Part of the local face, a tyndall scatter on the edge of an estuary, the first cousin of the color of the sea behind it, in a body the textbooks of the future will swear was impossible. Hold him for a second, then let the estuary go. Back in the warm room, the walls have returned. You and I are going to talk now about how that gene got there, how it traveled, how it spread, how a single mutation, in a single body, in a single generation, became a trait carried by roughly 800 million people alive on the planet today. To do that, we have to leave Western Europe and ride east across the Carpathian Mountains, down into the long flat grasslands of what is now southern Ukraine and Romania and the southern flank of Russia. You and I are going to step into a world that no longer exists. A world, you can almost smell it, of horse milk cheese and copper trinkets and timber wagons drawn by oxen. A world of pastoralists who buried their dead under low earth mounds called kurgans, who measured wealth in cattle, and who, around 5,300 years ago, began a westward migration that would reshape the entire genetic and linguistic landscape of Europe. These are the Yamnaya. The name, you may not have heard, comes from the Russian for pit grave because of those distinctive kurgan burials. Their territory ran from the lower Volga to the lower Dnieper, give or take, across the Pontic-Caspian steppe. Pontic meaning of the sea, because the steppe sits on the northern shore of the Black Sea and tapers eastward toward the Caspian. They herded cattle. They herded sheep. They almost certainly rode horses. You can picture the dust, although the timing of full domestication is still argued. They built wheeled wagons. They lived in temporary settlements that left almost no trace, and they buried their dead in mounds that, even now, you can walk among. The mounds are still visible from the air, scattered across the agricultural fields of modern Ukraine and southern Russia. Soft swellings in the flat grass like the breath of a sleeping animal under a blanket. There are hundreds of thousands of them. They were dug, slowly and with care, by people who believed something specific about what happened to the dead and who carried with them in the genome of every member of every clan an admixture they had picked up from earlier interactions with Western hunter-gatherers and with another deep population called the ancient North Eurasians. A small but real fraction of them carried, on chromosome 15, the derived allele at rs129138332, your allele. Perhaps most did not. They were, contrary to whatever you may have been told, predominantly dark-eyed and dark-haired, with intermediate skin tones. But the gene was there, in their gene pool, at low frequency, ready to travel and ready, eventually, to land in someone like you or someone you love. Around 3,300 BCE, by the standard archaeological dating you can find in any reference, they began to move west. The reasons are still debated: climate fluctuation, demographic pressure, the developing technology of dairy and wheeled transport that allowed them to cover ground at a speed previous populations could not match. The migration was not a single sweep. It was a long, slow, generational expansion, with successive waves pushing into the river valleys of Central and Northern Europe over the course of several centuries. They brought with them, into your mental map, a particular kind of pastoral economy. They brought with them, almost certainly, the Proto-Indo-European language family that would, in time, give rise to nearly every European tongue you have ever heard. And they brought with them their genes, which mixed with the genes of the populations they encountered and which, eventually, mixed into yours. The Western hunter-gatherers were still there, in pockets, although they had been displaced or absorbed by earlier Neolithic farmers from Anatolia. The Anatolian farmers were there, with their pale wheat and their painted pottery and their permanent villages. Across the river valleys of Europe, three deep ancestral streams, the Western hunter-gatherers, the Anatolian farmers, and the incoming Yamnaya, began to braid together into the populations that would, in time, become the Bronze Age cultures of Europe: the corded ware, the bell beakers, the early Indo-European speaking peoples of the 2nd and 1st millennia BCE. In every braid, you should picture the rs129138332 allele was carried somewhere in the strands. The Western hunter-gatherers were carrying it at high frequency in some places. The Yamnaya were carrying it at low frequency. The Anatolian farmers were largely without it. As the cultures admixed, generation by generation, the allele was reshuffled into new genetic backgrounds, perhaps your own. It found itself, sometimes, in skins that were lighter than the Mesolithic ones, because the skin-lightening alleles, which had only recently begun to spread, were now riding alongside it in some populations. It found itself, sometimes, in hair that was lighter than dark, because the hair-lightening alleles were spreading too, on their own evolutionary trajectories. The combination that the modern world thinks of as the standard Northern European phenotype, pale skin, light hair, blue eyes, was not a single package handed down intact from a single source. It was a recombinant, a braid of 3 different inheritance streams, layered over the course of 3,000 or 4,000 years of admixture. Cheddar Man was the eyes without the skin. The early Yamnaya were neither. The descendants of all of them, blended together in the populations of Bronze Age and Iron Age Europe, were the package the textbooks would later mistake for a single ancient inheritance. By the time the historical record begins, Greek and Roman writers describing the populations of the British Isles and Northern Europe, the trait map is already roughly in the shape it will hold into modern times. Tacitus writes about the blue-eyed Germani, Strabo notes blue-eyed Celts. The Romans, who tended toward darker eyes themselves, find the trait worth remarking on, in the way that a feature becomes worth remarking on only when it is not the local default. North of the Mediterranean, blue eyes are common. In the Mediterranean itself, where you may have traveled, they are rarer, although not absent. In the Near East and Persia, they appear here and there, often associated with the populations that had moved south from the steppe in earlier waves of Indo-European expansion. In Africa and East Asia and the indigenous Americas, they are essentially absent, except in cases of admixture or genetic disorders unrelated to RS129, 13832. The map is uneven. The map, the one you are watching with me, has always been uneven since the first founder generation. It was uneven in the Mesolithic. It was uneven in the Bronze Age. It is uneven now. The modern numbers, which the dossier lays out cleanly, are worth walking through slowly with you, not for the sake of the numbers themselves, for the sake of the picture they paint. In Estonia, blue eyes are present in approximately 89% of the population, the highest prevalence anywhere on Earth. In Finland, the figure is the same, give or take, perhaps in faces you have seen. In Sweden, the prevalence ranges between 78 and 90% depending on the region and the survey. In Iceland, it is approximately 75.5% a population shaped by Norse and Gaelic settlement in the 9th and 10th centuries, with a strong founder effect of its own layered on top of the original one. In Denmark, where Eiberg sits in front of you, the figure is between 60 and 64%. In the Netherlands, around 60 to 61. In Poland, around 52. In the United Kingdom and Ireland, somewhere between 42 and 50%, with the higher end in the north and west, where Celtic and Pictish ancestry layers thicker. In Germany, around 39 to 40. In the United States, the figure is approximately 27%, almost entirely a function of which European populations sent immigrants there in which centuries. A kind of demographic crystallography that you can read off the genome of a country with surprising precision. In Africa, the figure, you should know, is essentially zero except for isolated cases of admixture. In East Asia, the same. In the global population taken as a whole, the figure is between 8 and 10%, roughly 800 million people, give or take, alive on the planet right now. Walking around with eyes that scatter blue light because of one letter in one regulatory site that one ancestor, one night, somewhere near the Black Sea, 6 to 10,000 years ago, happened to carry into the next generation. 800 million is a number large enough to lose your sense of scale on. Try it differently. 1 in 12 people you have ever passed in the street, anywhere in the world, has eyes that are descended from that single founder. 1 in 12, not 1 in 12 in your country. 1 in 12 on the entire planet. Some places more, some places less, but globally, 1 in 12, perhaps you, perhaps the person beside you. Every 12th pair of eyes you have ever met, on any train, in any airport, on any walk, traces in a chromosomal sense back to the same person. It is a humbling little fact, in the way that genealogy is humbling when you sit with it long enough. The 12 people on your street with blue eyes are, all of them, distantly your cousins through that line. The girl in the supermarket with pale eyes and dark hair, who looked up briefly when you walked past your cousin, in this small chromosomal sense. The man on the train with eyes the color of weak tea and a sliver of grey at the temples, who was reading a book, your cousin. The child who pressed her face against the window of the bus and watched the rain, your cousin. 800 million cousins, distributed thinly across the planet, descended from one body, in one place at one time. All of them carrying the same haplotype on chromosome 15, all of them quietly proving the founder effect every time they look at anything in a bright room. The mathematics of how this happened, how one mutation, in one person, became a trait carried by hundreds of millions, is the question that opens the next part of the story. It is also the question that, even in 2026, even with all the genome sequencing in the world, the field cannot definitively answer. The mutation is settled. The mechanism is settled. The founder effect is settled. The geographic origin, you should know, is reasonably constrained, with a few honest caveats. But the question of why the allele spread, Why it grew from one carrier to roughly an eighth of the human population is genuinely, professionally, openly unresolved. It is one of the longest open questions in human evolutionary genetics, and the people who have spent their careers thinking about it disagree, in good faith, about which of several explanations is most likely. You and I are going to walk through their arguments now. You and I are going to give each one its fair weight. You and I are going to refuse to pick a winner. The honest answer, and the answer the dossier insists on, is that the science is genuinely contested. The evidence is split. Researchers argue. The consensus is weak. You and I will hold all of those positions in the room at once. Walk first, with me, into a small university office in Brisbane, Australia. The window is open. Subtropical afternoon light comes through, warm and slightly heavy on your skin. There is a desk piled with genetics journals, the way only an academic's desk can be piled strata of paper that record the order in which the papers arrived. There is a man at the desk in his 60s, with reading glasses pushed up to his forehead and a coffee that has gone cold. His name is Rick Sturm. He is one of the leading pigmentation geneticists in the world. He is writing a paragraph. You can lean in if you want. That will, in time, appear in a review article. The paragraph begins. There is no physiological evidence that lighter irides confer any optical advantage in low light conditions. He is writing this paragraph because, for most of the 20th century, the field assumed that they did. He is writing this paragraph because that assumption was wrong. The assumption was the vitamin D hypothesis, and for decades it was the dominant explanation for why blue eyes spread. The thinking went like this. As humans migrated out of Africa and into the higher latitudes of Europe, they encountered a sun that was weaker, and a sun that was weaker for longer parts of the year. UVB radiation in the upper layers of the atmosphere is what drives the synthesis of vitamin D in human skin. Vitamin D is essential for bone development, for calcium absorption, for the prevention of rickets, and a long list of other deficiencies that show up vividly in skeletons and dimly in soft tissue. Darker skin filters more UVB, lighter skin filters less. So at high latitudes, where the UVB is already scarce, lighter skin carries a survival advantage by allowing more efficient vitamin D synthesis. The textbooks bundled all the lighter pigmentation traits, light skin, light hair, light eyes, into a single co-adapted package, and explained their distribution by reference to a single selective pressure. Lighter at the top of Europe, darker at the bottom. The map made sense. The story was clean. Generations of biology students, perhaps you among them, learned it as the answer to why blue eyes had spread. The story has not survived the ancient DNA. The Mesolithic Western hunter-gatherers Cheddar Man, Labranya I, Loshbor Man, the Motala hunters broke it. They had blue eyes. They had dark skin. They lived at high latitudes for thousands of years with that combination, getting by on whatever vitamin D they could synthesize through their dark skin. Their diet supplied the rest, from oily fish and animal fat and lichens and the few wild plants they gathered. Their iris color, like yours, did nothing for their vitamin D, because the eye is not a pigment synthesis organ. The eye is a sensor. It does not absorb sunlight, you should know, in any way that contributes to internal biochemistry. Whatever happens in the iris, in terms of pigment, has zero downstream effect on the production of vitamin D anywhere in the body. Sturm has been making this point in print for years. He makes it again in the paragraph he is writing in Brisbane, in the warm afternoon, with the cold coffee. There is no physiological mechanism by which a blue iris would assist vitamin D synthesis. There never has been. The eye is not a skin patch. The eye does not metabolize the sun. The vitamin D hypothesis, as an explanation for why blue eyes specifically spread, is not a plausible story. It explains the spread of light skin reasonably well. It does not explain the spread of light eyes at all. That leaves the field with a puzzle. The mutation is real. The spread is real. The selective advantage is, on inspection, weak or absent. So why did it spread at all? Several alternative hypotheses have been proposed. None of them are decisively confirmed. We will walk through each of them slowly. Settle in further. The night is long. There is no rush. The first alternative, and probably the most discussed one in popular science writing, is sexual selection. The argument runs like this. In a Mesolithic population, where every adult had brown eyes, a single individual born with the derived G genotype would have stood out visually. Not just stood out, would have been arresting. The Tyndall blue is striking even in modern populations where blue eyes are common. In a population where they had never been seen before, the impact would have been disproportionate. Striking traits, the argument continues, can be amplified rapidly by preferential mating. If the visually unusual individual was preferred as a mate, even slightly, by some fraction of the population, the allele would have been overrepresented in the next generation relative to its starting frequency. Over a few hundred generations, with even a small mating advantage, the allele could have grown from a single founder to a substantial fraction of the population without ever conferring a survival advantage in any classic sense. The mechanism is sometimes called, informally, the Paul Newman effect, after the Hollywood actor whose blue eyes were used in arguments by some evolutionary biologists in the late 20th century to illustrate the idea. The serious version of the argument runs through the work of Charles Darwin himself, who first proposed the idea of sexual selection as a driver of striking traits in the descent of man, and through the work of Ronald Fisher and many others in the 20th century who modeled the conditions under which a sexually selected trait can run away from its starting frequency. Sexual selection has elegant features as an explanation. It does not require the trait to be useful. It does not require the trait to be adaptive in the conventional sense. It only requires the trait to be preferred. It is also self-amplifying once a critical mass of individuals in a population carries the trait. The preference for the trait becomes part of the local mating environment, and selection presses harder on its own. The maths can produce very rapid spread under the right conditions. And it has a certain narrative pleasure to it. The first blue-eyed child, somewhere near the Black Sea, 6,000 to 10,000 years ago, born to brown-eyed parents through some combination of new mutation and chance recombination, looked up at a world that had never seen those eyes before, and the world looked back, and something in the way it looked back tilted the chances of that child's genes ending up in the next generation by a fraction of a percent. Repeat that fraction of a percent for 200 generations, with population growth and migration on top, and you arrive at modern Europe. It is a story that fits the genome. It is a story that fits the timing. It cannot be definitively proved, because sexual selection is not the kind of force that leaves an unambiguous footprint in the genome its signature looks. In many ways, like the signature of drift, but it cannot be ruled out, and many serious evolutionary biologists hold it as the most likely explanation. The second alternative is closely related, and in some accounts is folded into the first one. It is the neoteny hypothesis. The argument goes, human infants are often born with blue or grey eyes that darken in the first 1 to 3 years of life, as melanin gradually deposits in the iris stroma. The combination of certain features that we associate with infancy, large eyes, soft features, a particular cranial shape, a particular skin tone, blue or grey eye color, triggers caregiving and protective responses in adult humans. We are wired to find infants attractive in a non-sexual way. Some of those responses are relatively easy to study. Babies are visually arresting to adults. Most adults will turn their head when a baby cries. Most adults will soften their voices when a baby is in the room. The neoteny hypothesis suggests that some of those responses bleed into adult mate selection. adults who retain certain infant-like features into adulthood, including, possibly, permanent blue eyes may trigger weaker versions of the same protective and care-related responses, and that those weaker responses may be enough to bias mate selection toward the more neotenous individual. If permanently blue-eyed adults trigger a flicker of the same warm response that babies do, even in tiny degree, the allele could be amplified in the same way that the sexual selection argument proposes, by a mechanism that is partly sexual and partly something more diffuse than sexual. The neoteny hypothesis is hard to test directly. It is hard to ask the relevant questions about a population that no longer exists. And it is hard to disentangle which traits in modern populations are genuinely neotenous in the relevant sense, and which are simply things we have learned to find attractive through cultural exposure. But the broad argument that traits associated with youth, when retained into adulthood, can carry a small mating bias, is well established in evolutionary biology, and it is at least a plausible piece of the puzzle. Sturm and others have noted, in print, that the neoteny case may be partly responsible for the persistence and spread of blue eyes, even if it cannot fully explain the speed and scale of the spread on its own. The third alternative is the one Sturm himself proposes most strongly in his recent writing. It is the seasonal affective disorder hypothesis. The argument here is more behavioral and biochemical than the previous two. Sturm has argued, drawing on a body of research that links eye color with neurochemistry in modest but real ways, that lighter-eyed individuals may be slightly less susceptible to the depressive symptoms of long northern winters than darker-eyed individuals. The mechanism, if it is real, is not optical. It is not that more light reaches the retina through a less pigmented iris in any meaningful quantity. The mechanism is hypothesized to be neurochemical. There may be a correlation between the genetic background that produces the blue iris and the genetic background that influences serotonin or melatonin processing or some other downstream regulator of mood and energy. If lighter-eyed individuals at high latitudes were even slightly less likely to suffer the worst seasonal depressions, they would have been more active hunters and gatherers in the long winter months, more reliable parents, more effective members of small tribal communities at exactly the times of year when those communities were under the greatest stress. The fitness advantage need not be large to produce the spread we observe. It only has to be there. The SAD hypothesis is harder to test in ancient populations. For obvious reasons, depression does not fossilize. It is also controversial, even among Sturm's colleagues, because the correlation between eye color and neurochemical variation, while real in some studies, is small in absolute terms, and may be confounded by the fact that lighter-eyed individuals tend to come from populations that have had longer to adapt to high-latitude life in many other ways. Sturm has continued to argue for it, partly because he believes the optical version of the argument, the vitamin D hypothesis, is dead, and partly because he thinks the genetic linkage between pigmentation and neurochemistry deserves more attention than it has received. The case is not closed. The hypothesis is on the table. It may turn out to be partly right, or fully wrong, or right in a different way than its current formulation predicts. The fourth alternative is the most boring one, and possibly the most powerful. It is genetic drift. Drift is the technical name for the way allele frequencies fluctuate randomly from one generation to the next, especially in small populations. By virtue of the simple fact that not every individual reproduces, and not every reproductive event passes on the same set of alleles. In a small Mesolithic tribe of, say, 200 people, the random fluctuations in who has children and how many can shift allele frequencies by a few % per generation, just by chance. Across thousands of years and many small populations, drift can amplify some alleles to high frequency in some places and eliminate them entirely in others, with no selective pressure required at all. If the founder of the RS129-13832 mutation lived in a small, geographically restricted population, and if that population happened to undergo a rapid expansion in the centuries after the mutation arose, which the archaeological and genetic record suggests it did, several times, then drift alone, combined with population growth, could account for a substantial fraction of the spread. Drift does not need a story. Drift does not need a reason. Drift is just what populations do when they bump along through demographic time. The argument is that the spread of blue eyes may have been substantially driven by demographic accident, the right founder, in the right population, at the right time. Going through the right expansions in the right centuries would carry the allele into the modern world without any selective help at all. Drift is unsatisfying as a narrative because it does not give you a story. It gives you noise. But noise is, in many cases, the correct answer in evolutionary biology. And a great deal of human variation is best explained by some combination of drift and demographic history, rather than by any specific selective story. The honest position, according to the dossier and to a growing number of researchers, is that the spread of RS129-13832 was probably driven by some combination of all four of these mechanisms: sexual selection, neoteny, possibly a small SAD-like fitness advantage, and a substantial dose of drift and founder effect amplification through the demographic upheavals of the Neolithic and Bronze Age. None of them, alone, fully explains the pattern. All of them, together, plausibly do. The science is not settled. The evidence is split. Researchers argue, in good faith. The consensus is weak. The mutation is settled. The mechanism is settled. The geographic origin is reasonably settled. The reason it spread is not. Sit with that for a moment. Most stories in popular science end with an answer. Most evolutionary explanations of common traits offer a single mechanism, a single cause, a single arc from origin to outcome. This one does not. This one ends with a shrug, an honest one, by serious people who have spent their careers thinking about it. It is one of the things that makes the case interesting. It is also one of the things that makes it sleep appropriate. In a way, the universe does not always tidy itself up. The genome remembers things it cannot fully explain. The eye in the lightbox is the eye in the lightbox. Whatever the reasons for its presence, the reasons can wait until morning. The eye is here now. It has been here for thousands of years. Let Sturm finish his paragraph. Let the warm Brisbane afternoon thicken. The window is still open. The journals on the desk hold the arguments down with their weight. We are going to walk away from his office now, slowly, back across the world, back through the centuries, back through the Bronze Age and the Yamnaya and the dark-skinned Mesolithic hunters and the Neolithic transition, back to the moment the mutation first appeared, in one body, somewhere near the northwestern Black Sea, between 6,000 and 10,000 years ago. We are going to spend a little time there, in the shallow of that moment, before we return to the room. Walk into the moment. There is a clearing in a forest near a river. The river, in front of you, is narrow and slow. The trees are mixed deciduous, oak and ash and lime, the kind that filled the Pontic-Caspian fringe before the steppe took over the deeper interior. The leaves are turning. There are men and women in the clearing, working at hide, scraping a deer hide with a flint blade, the smell of smoke and grease and damp wool. There is a fire, and you can almost feel its warmth. The fire is small, contained in a ring of stones. There is a child sitting near the fire, 3 or 4 years old, watching the women work. The child has eyes that her mother has not seen in any other child she knows. The eyes are light, not pale grey, not light hazel, not the darkening transitional eyes of an infant whose melanin has not yet come in. The eyes have stayed light. The mother does not know why. There is no language available to her to ask the question. There is no one in the camp who has eyes like that. There is no story in the local stories about anyone who ever had eyes like that. The child is, Quite simply, the first one. She will not know that she is the first one. She will go on to have children of her own, in time, and some of those children will inherit the dialed-down regulatory site that her own body somehow assembled at conception. And those children will pass it on, and some of those grandchildren, in turn, will pass it on to the rare partners who happen to carry the same allele on the corresponding chromosome. And over the centuries, one mating event at a time, the haplotype around chromosome 15 will spread. We do not know that the founder was a girl. We do not know that she lived in this exact kind of clearing. We do not know what her name was, and we will never know. Because the language her people spoke is gone, and the names they used are gone, and the stories they told around fires like this one are gone. Lost like all the stories of all the small populations that gave rise to all the long inheritances of the modern world. What we know is that she existed, or someone like her did. The genome does not lie about descent from one. The chromosomal readout in the lightbox in Copenhagen is, in a strict and verifiable sense, a long letter from her, written in the language of inheritance, copied through every body in every generation between her and the present, arriving here, tonight, in our hands. She could not write. She had nothing to write with. The letter was written anyway, by the only mechanism nature had the duplication of DNA, every generation, in every cell, in every lineage that survived to reproduce. 800 million carriers later, the letter still reads. Watch her for a moment longer. The fire is small. The smoke goes up vertically, in front of you, into a still afternoon. The women keep scraping the hide. The men come back, in your line of sight, from the river with fish. The child gets up and follows her mother somewhere, the way 3-year-olds do, distracted by something on the ground. The forest, around you, is quiet apart from the work and the fire and the distant call of crows. None of these people know what you now know, that they are at the beginning of anything. None of them know that one of the children running between them carries a single letter on chromosome 15 that will ride the migrations west into Europe over the next 4,000 years, and east into Anatolia and the Caucasus over the next 2,000, and across the Atlantic in steerage in the 19th century, and around the world in jet aircraft in the 20th, and into the lightbox in Copenhagen in the 21st. They do not know, the way you know, that the child is the founder. The child does not know that she is the founder. The founder is just a child by a fire on an ordinary afternoon, near a river that no map of the time records. In a forest that has long since given way to grain fields and roads and the slow advance of agriculture into the Pontic shore. She lived, and she had children, and her children had children, and the line did not break, and that line, in some small way, may have led to you. That is the only requirement. That is the only thing the genome asked of her. She met it. Let the clearing fade. Let the fire smolder down. The forest holds the smoke for a moment, and then a wind moves through the leaves, and the smoke disperses, and the moment is gone. We will not be able to return to it. No one will. The river will silt up over the millennia. The clearing will be ploughed and re-ploughed and finally tarmacked over. The bones of the founder, wherever she was buried, will dissolve back into the soil under the slow work of time and water and roots. The genome alone will survive. The single letter on chromosome 15, repeated faithfully for some 400 generations, will be the only physical trace of her that lasts. Most of what we are, when we look closely, is exactly that kind of trace, a few letters from a few founders. Repeated and recombined, the surviving fragments of the lives of strangers we will never meet. The blue eye is one of the more visible traces. Most of them are invisible. They live inside us, in the long script of the genome, doing the small jobs they were given by the founders who first wrote them. Sleep with that for a moment, if you like. You are a library of strangers. You always have been. Now, the dossier is careful to flag a few myths that have grown around this story, and we should walk through them so the picture in your head does not absorb anything that does not belong in it. The first myth, perhaps you have heard it, is that blue eyes are dying out. You may have read this. It is repeated often in casual articles and in the kind of evolutionary fluff that turns up in magazines twice a decade. It is wrong. Recessive alleles do not die out simply by being recessive. They hide. They sit in heterozygous carriers who do not express the trait themselves, but who pass the allele on to half their children. Hardy-Weinberg equilibrium, the basic mathematical framework of population genetics, predicts that in a stable population, the absolute number of recessive alleles will remain stable across generations, regardless of how many homozygous individuals are present in any one generation. The visible expression of the trait can dip and rise, depending on mating patterns and demographic shifts, but the alleles themselves are conserved. The genetic record we see today shows blue eyes at a roughly stable global frequency over the last several hundred years. The trait will not vanish in our lifetimes or our grandchildren's lifetimes, including yours. The articles are wrong. The genome conserves what it has. The second myth is the inverse, that all blue-eyed people are essentially close family. This is partially true and substantially misleading. They share, yes, a single genetic founder for the RS129-13832 mutation. The chromosomal haplotype around it is the same across all of them, which is the strongest possible evidence of a single source. But the rest of the genome is enormously diverse across blue-eyed people, the same way it is enormously diverse across any large modern human population. Blue-eyed people share one chromosomal segment. They do not share most of the rest of their DNA. The shared inheritance is real and small and localized. It is not the kind of shared inheritance that makes a family. It is the kind of shared inheritance that makes a single allele history. In addition, the dossier reminds us that pedigree collapsed the mathematical fact that everyone alive shares common ancestors going back enough generations. Means that every human alive today is descended from common ancestors who lived around 10,000 years ago, regardless of eye color. The blue eye case is unusual not because the descent happened, but because the specific haplotype survived intact and unambiguous, readable, right to the present. The descent itself is universal. The visible signature of it, in a single allele, is what makes the blue eye case remarkable. The third myth, perhaps the one you remember, is the high school version of eye color genetics. Brown is dominant, blue is recessive. Two blue-eyed parents will always have blue-eyed children. Two brown-eyed parents may have brown or blue children depending on whether they carry the recessive allele. The Punnett square will tell you everything. This is the version most of us were taught, and it is, like most of high school biology, a clean teaching simplification of a messier reality. The OCA2, HERC2 axis, is the largest single contributor to eye color, and within that axis, the RS129-13832 polymorphism does behave roughly recessively. But human eye color is polygenic 16 genes or more, contribute by current count, with new candidates being identified as the genome is studied more closely. There are modifier genes that can produce intermediate or unexpected outcomes. Two blue-eyed parents can, occasionally, have a brown-eyed child if the modifier genes on either side combine in the right way to push pigment back into the front stroma. It is rare, but it is genetically possible, and it has been documented in published cases. The Mendelian rule is not wrong. It is just incomplete. The genome is messier than the textbook suggests. The fourth and final myth in the dossier is heterochromia, the condition where a person has two different colored eyes, or sectoral patches of different color within a single eye. It is sometimes assumed to be a mixing of pigments, the way mixing two paints produces a third color. It is not. Sectoral heterochromia is a localized genetic mosaicism or somatic mutation in which the regulatory site of OCA2 is functioning normally in one section of the iris, and is suppressed in another. The brown patch is brown because melanin is produced there. The blue patch is blue, you should know, because melanin is suppressed there, and structural scattering takes over. The two colors are produced by the same physics that produces the colors in normal eyes. They simply happen, you can imagine, to be expressed in different patches of the same iris. The condition is rare, but it is a beautiful illustration of how local the regulatory control of pigment is, and how the iris is not a single homogeneous surface but a patchwork of cells, each making its own decision in response to its own regulatory inputs about how much pigment to lay down. Settle in further. You and I are going to walk through one more set of facts before we turn toward the closer. These are the medical correlations of blue eyes, the practical, biological consequences of carrying less melanin in the front of the iris than the average human eye carries. They are not central to the spread story. They are not central to the evolutionary debate, but they are worth your knowing, and they round out the picture. The first is light sensitivity. Melanin in the iris stroma absorbs incoming light, especially the higher energy short wavelengths in the blue and ultraviolet range. A blue iris, perhaps yours or someone you love, by definition, has less of that absorbing pigment. More high-energy light passes through the front of the iris and into the deeper structures of the eye. Over a lifetime of exposure, especially in environments with high ambient sunlight, this has measurable consequences. Studies have documented roughly a twofold higher incidence of age-related macular degeneration in blue-eyed individuals compared to brown-eyed individuals, though the association becomes less clear-cut when controlled for broader genetic ancestry. The mechanism is reasonably well understood. High-energy visible light, especially blue light, contributes to oxidative damage in the macula over time, and lighter irides emit more of it. It is one of the very few cases where the lack of melanin in the front stroma has a directly demonstrable downstream effect on health. The second is the slightly higher risk of uveal melanoma cancer of the pigmented tissues of the eye in lighter-eyed populations. The risk differential, you should know, is smaller than for AMD, but it is real, and it follows the same logic. Less pigment in the front of the iris means less UV protection for the deeper structures. The ocular melanocytes, which can become cancerous when damaged, are slightly more exposed in lighter eyes than in darker ones. The absolute risk remains low, and uveal melanoma is a rare cancer, but the relative risk in blue-eyed individuals is higher than in brown-eyed ones, and it has been documented in epidemiological studies across multiple populations. The third is the common observation that blue-eyed individuals are slightly more sensitive to bright light in everyday situations. Many blue-eyed people report squinting. Perhaps you have noticed it in someone close to you, in conditions where brown-eyed people are comfortable. Many blue-eyed people prefer sunglasses, again perhaps familiar to you, in conditions where brown-eyed people do not feel they need them. This is partly a matter of subjective comfort, and partly a matter of the physical reality that more light is reaching the retina through a less pigmented iris. It is a small, mostly cosmetic point, but it is a real one, and it links the founder mutation to the daily lived experience of every person who carries it. The color you call your eye color is also a comfort threshold, in good light, in summer. There is one more biological consequence that is worth mentioning, and it is the one we already touched on briefly when discussing the color of newborn eyes. Many human infants are born with blue or gray eyes that darken in the first 1 to 3 years of life. The mechanism is not the RS129133832 switch. It is the fact that melanin production in the iris stroma is partly stimulated by light exposure. And infants in utero have not yet had the kind of exposure that triggers significant pigment deposition. After birth, the cells of the stroma begin laying down melanin, and the eye color shifts toward whatever the genetic instructions ultimately specify. Children who carry G at RS129138832, both copies of the chromosome with the suppressed regulatory site, never deposit enough melanin to obscure the structural blue, and the eye stays blue or grey for life. Children who carry A, or who carry one A and one G in heterozygous fashion gradually develop the brown or hazel that their genes specify, sometimes over months, sometimes over years. The slow darkening of an infant's eye is the single most accessible, observable demonstration of how this whole system actually works. You can watch it happen in real time, in real children, over the first 3 years of their lives. The genetics is not a hidden thing. It is a thing you can see in the eyes of every baby you have ever known. Hold all of that lightly. Most of the practical biology is small. It is the kind of small that you carry through a lifetime without ever thinking about, until you read a paragraph somewhere and recognize the pattern in yourself. Most of what makes us interesting, biologically including you, is small in this same way. The body holds the founder mutation. The founder mutation does its quiet work. The work shapes the color of an iris, and the color of the iris influences how a person experiences sunlight and bright rooms and the slow accumulation of oxidative damage over decades. None of this changes the larger story of who you are or what you do with your life. It only adds a small note to the margins of the page in the language of the founder, signed by a stranger you will never meet. We are nearing the end now. The night has gone deeper. The room has gone darker. We have walked a long way, you and I, from a basement in Copenhagen with a lightbox and 2 iris photographs, through a lecture theatre with a slide of chromosome 15, into a cave in Somerset where a labourer's pickaxe caught against a 9,000-year-old skull, along the Atlantic estuaries of Western Europe where dark-skinned, blue-eyed hunters walked for thousands of years, eastward across the Carpathians into the long flat grasslands of the Pontic-Caspian steppe, into the shallow of one quiet afternoon in a forest clearing where a single child was the first person on the planet to look at the world through the new color, then forward through the Yamnaya migrations and the Bronze Age admixtures, through the modern population genetics that maps the trait at 89% in Estonia and 27% in the United States, and 8 to 10% globally, through the long debate about why the trait spread, through the medical correlations and the myths, and the small biological consequences, all the way back to here, to the room you are in, to the bed or the chair you are sitting in, to the breath you are breathing, slowly, while the night holds its position around you. The world the founder lived in is gone. The forest she sat in is gone. The river she lived beside, the one you walked toward earlier, is silted up or rerouted or buried. The fire she watched in her mother's hands, the one you saw with me, has been ash for 10,000 years. The clothing, the language, the songs, the practices of her people are gone. Every name, every voice, every face, gone. What survives is the letter on chromosome 15, repeated through some 400 generations of strangers, copied with extraordinary fidelity by the cellular machinery of every body that has carried it, arriving in the present unchanged. 800 million people, perhaps yourself among them, now carry the same letter she carried. They walk through cities she could not have imagined. They board aircraft she could not have conceived of. They look out of windows in apartments above streets, above sewers, above buried rivers, and they look at one another, and they look at strangers, and they look at the people they love, and somewhere in their irises, a small Tyndall scatter is doing the same trick it did in hers, letting the long wavelength sink into the dark layer at the back of the eye, sending the short wavelengths bouncing back out toward the world. The trick is the same. The physics is the same. The single letter is the same. The carriers have moved on across the planet in numbers she could not have begun to count. She existed, or someone like her did. The genome insists on it. The lightbox in Copenhagen confirmed it. The math of pedigree collapse and founder effects, the one you are now familiar with, requires it. There was, somewhere, in some body, between 6 and 10,000 years ago, near a sea that still carries the same name, a single moment of inheritance that started a line that has not broken since. That line runs through every blue-eyed person on Earth, perhaps including you. It runs, in its quietest sense, through every brown-eyed person too, because pedigree collapse means we all share common ancestors going back 10,000 years. But in those people, the founder's specific letter on chromosome 15 has been overwritten by other inheritances in the same regulatory site. The letter survives where the regulatory site is left alone. The letter is overwritten where another lineage's contribution has covered it up. The genome holds many letters from many founders. This particular letter, perhaps yours, is held by approximately 1 person in 12. That is what the science says. That is what Eiberg confirmed. That is what every ancient genome from every Mesolithic cave has continued to confirm for you. That is what the Yamnaya kurgans and the corded ware admixtures and the Bell beaker spread continued to underwrite. That is what the modern population maps continue to display when you read them. 1 mutation, 1 ancestor, roughly 10,000 years, roughly 800 million carriers. The numbers do not decide anything for you about meaning. The numbers do not tell you what to think about your own eyes, or your children's, or your partner's, or the eyes of the person who will pass you on the street tomorrow. The numbers only describe, when you sit with them, the shape of an inheritance. The meaning, if you want one, is yours to fashion. The genome only does one thing. It remembers. It has been remembering this particular thing, faithfully, since long before any of us could speak. It will keep remembering it, with luck, long after we have all stopped speaking. The single letter on chromosome 15 does not require our attention to do its work. It does its work either way. The eyes that scatter the light scatter it whether we are looking or not. There is a quietness, perhaps you can feel it now, that settles into a room when a long story finishes. You have probably felt it before. The voice stops, the page closes, the lights are still on, or they are off, but the quality of the air has shifted. The stories that were inhabiting the room have receded back into the wallpaper, back into the books on the shelf, back into the corners. The room becomes itself again. The pillow under your head is itself. The blanket is itself. The breath is the breath. The ground beneath you is solid and still. Whatever you were thinking about while you listened, the lightbox, the cave, the clearing in the forest, the founder, her child, the letter on the chromosome has receded, and what is left is the room. The room is warm. The room is quiet. The room is yours. Your blanket is warm. The fabric of your pillow is soft against your face. The temperature in the room is exactly what you set it to, or what the building's thermostat decided, and your body has adjusted to it without you needing to think about it. Your breathing has slowed. It is slower now than it was when we started. You may not have noticed that. The rate of your breath drops a little when a voice continues evenly for a long time, and the muscles in your face soften. And the small tightnesses across the shoulders and the jaw and the ribs let go a fraction at a time. None of this requires effort. None of this requires you to do anything. Your body knows how to settle into a long voice. The body has been settling into long voices, around fires, in clearings, in caves, in tents, on the steps, in basements in Copenhagen, in lecture theatres, in offices in Brisbane, for as long as there have been bodies and voices. The founder, in her clearing, would have known the same kind of settling. Her child would have. Her child's child would have. The settling is one of the longest inheritances of all, older than any single allele, older than chromosome 15, older than the species itself. The body knows what to do. Let it do it. Your eyes are heavy. They may already be closed. If they are, that is fine. If they are not, that is also fine. Whatever color they are, The color is doing its quiet structural work, the pigment absorbing or the stroma scattering, the back of the eye holding its dark layer steady. The long wavelengths sinking into the dark, and the short wavelengths bouncing back, and the work continues with or without your attention. In near darkness, the color is barely visible anyway. Brown eyes, in low light, look black. Blue eyes, in low light, look grey. Hazel eyes look indeterminate. The color drains out of every iris when the ambient light drops below the threshold the structure needs to do its scattering. In the dark, every eye, including yours, is the same eye. Every iris, including yours, becomes a dark circle around a darker pupil. The thing that distinguishes us in daylight, the thing the founder's mutation introduced into the human face 6,000 to 10,000 years ago, recedes when the light goes. It comes back in the morning. For now, in the dark, we are all one population again. Eye-wise, the color is gone. The structure is still there. The pigment is still there. They are just doing their work where no one, not even you, can see them. The lightbox in Copenhagen is dark now. Eiberg went home long ago. The 2 iris photographs are still in the lab, somewhere, in a folder, in a drawer, with the dust accumulating on their edges. The chromosomal readout tape beside them has yellowed slightly with age. The basement is empty. The fluorescents have been turned off. The centrifuge has been switched off. The hum that ran through the room while he worked, the one you could almost hear earlier, has gone with the rest of it. If you were to walk down those stairs now, in the middle of this night, you would find the lightbox cold, and the photographs invisible in the dark, and the readout indistinguishable from the wall behind it. The whole apparatus of scientific discovery, when no one is using it, is just furniture, and you are not using it now. The discovery has moved on. It lives now, in the genomes of every person who has ever read the paper, and in the genomes of every person whose eye color the paper explained. The room itself has become quiet. The work is finished, for tonight. Tomorrow, someone will turn the fluorescents back on, and the centrifuge will start up again, and the basement will fill again with the small hums of normal scientific life. For now, it is dark. The forest the founder sat in, the one you walked through earlier, is dark too. It always was, at this hour. The fire she watched in her mother's hands burned down many thousands of years ago. The clearing reverted to woodland, and then to grassland, and then to ploughland, and then to whatever the modern landscape of that part of the Black Sea coast happens to be. The trees came and went. The river moved. The ground shifted, slowly, the way ground does over millennia. The founder's bones returned to the soil. Her skin returned to the soil. Her hair returned to the soil. Everything physical returned. Only the letter survived. The letter is in your room now, possibly, depending on which set of irises is closing somewhere in the dark beside you, or in your own face, or in the face of someone you love. The letter is everywhere, and the letter is nowhere. It is a single base on a single position of a single chromosome, repeated through every cell of every carrier, doing its small regulatory work without fanfare, without ceremony, without any awareness on its own part that it is one of the longest single-source inheritances in the entire history of the species. The letter does not care, in the way you do not need to care now, that it is famous. The letter does not care that you and I have just spent 2 hours discussing it. The letter simply continues. It continues in you. It continues in the 800 million. 800 million is a lot of cousins to share a room with. You will not meet most of them. Most of them will not meet each other. The line that connects you to all of them runs through a single body, in a single place, at a single time. And after that body it fans out into all the demographic complexity of all the millennia between then and now, and lands here, on this night, in this room, in the closing minutes of a long voice. Whether your eyes carry the letter or not, the line still runs through your pedigree. Pedigree collapse is generous like that. Everyone shares everyone, far enough back. You are descended from her either way. whether the chromosome 15 haplotype came down to you intact, or whether it was overwritten by other inheritances in your lineage. You and she are connected by the slender thread of common descent that connects every human alive to every other one. You are descended from her. You are descended from her child. You are descended from her child's child. The thread does not break. The genome has been faithful to it, in some chromosomal stretch or another, for the entire 10,000 years. That is the story, or that is the shape of the story we know how to tell. The full story is messier, of course, and there are loose threads that we have already acknowledged, the ancient North Eurasian molecular precursors that complicate the strict 10,000-year window, the unsettled debate about why the alleles spread, the small caveats around the global distribution numbers, the continuing work of the field as new ancient genomes are sequenced and new modifier genes are identified. The story is not closed. The science is not finished. There will be more papers, in more journals, in more decades, refining what we know and occasionally upsetting what we thought we knew. The shape of the story will change in small ways, the way all good stories change in the hands of better evidence, but the core will hold. One mutation, one founder, approximately 10,000 years, approximately 800 million carriers. The lightbox in Copenhagen confirmed it. The Mesolithic genomes confirmed it. The modern population maps continue to display it. The story holds. The night holds too. It has been holding all along. Outside the room, wherever you are, the dark continues. Whatever clouds or stars are above the building you are in, whatever rain or wind or stillness, the night is doing its work. The day will come back. In the time it always comes back. For you, the room around you will fill again with whatever light the morning brings. Your eyes, whatever color they are, will catch that light and scatter or absorb it according to the structure they have inherited, and you will go on with the day. The founder's letter does not require any of this. The letter is patient. It has been patient for 10,000 years. It will be patient for tonight. Every eye in the dark is the same. The breath is soft. The light box in Copenhagen is dark. The lab empty. Sleep well.